Okay, thank you, Jeremy, for that introduction. And thank you again to the speakers that we heard this morning. You really provided an excellent introduction to what I'm going to be sharing with you. We heard about multiple strategies to essentially manipulate and harness the strength of the immune response. And what those strategies have in common is they all require an understanding of what it is the immune system recognizes so that that can be exploited and enhanced. So I'm going to be sharing our experience in working with groups to help them understand and define what it is the immune system recognizes. Okay, so here we have the cartoon on the far right detailing a class 1 antigen processing pathway. I think we're all very familiar with that, so I'm not going to be going through that. But by being able to define what those epitopes are that are being presented via MHC class 1, this will help to improve the specificity of your immunotherapies and vaccines, as well as to reduce the lead time in their development. And so I'm going to be sharing some of our technologies for epitope discovery from this class 1 pathway, as well as providing tools on how to then track immune responses against those epitopes. Okay, so there are several different platforms for epitope discovery and immune monitoring. And what we at ProImmune will do is help to identify the best approach moving forward based on the objectives of your program as well as the resources that you have available to you. And what I mean by that is whether you're working with clinical samples, maybe derived from a novel cancer vaccine, or if you just have a sequence from a virus, for example, that you want to further characterize, we'll be able to help you to make those decisions. Now, there are several different platforms for epitope discovery and immune monitoring, and we heard about a few of those earlier this morning. I am going to be focusing on the antigen presentation and the MHC-peptide binding assay as tools for epitope discovery, but I do have a couple others listed here just to highlight briefly. In silico prediction algorithms are often used by groups as a first pass to help to narrow down that antigenic focus. The class 1 prediction algorithms like IEDB and CIFPython are generally pretty good. They're also free and they don't require the use of any material. But of course there are limitations as these are predictive tools. Functional assays like T-cell ELISpots and ICS can also be used for epitope discovery, and we heard a little bit about that this morning. These you would take in a matrix-style approach using peptide pools to determine hotspot regions from your antigen, and then go through additional rounds of analysis to determine the minimal epitope. So it can take several rounds of analysis and be very cell-intensive. So I am going to be talking about T-cell ELISpots today in pretty great detail, but more so in the context of an immune monitoring tool. I'm also going to be discussing our multimers, our Pro5 Pentamers and Pro-T2 Tetramers for detection of antigen-specific CD8 and CD4 T cells by flow cytometry. Okay, so the first assay that I will focus on is our ProPresent Antigen Presentation assay, and this is used to determine the naturally processed and presented epitopes from a protein of interest. Now, this is a very powerful tool in that it combines the highly sensitive nature of proteomics with immunology. So we can look at both class 1 and class 2 peptides using this method. I'm going to be talking about the class 1 approach. You'll hear more about class 2 tomorrow and exogenously loaded proteins from Jeremy's presentation. Now for the class 1, this is quite a rapid turnaround. Once we receive your cells, it takes about 2 to 3 weeks before we generate the report. And we have a lot of experience in this area, working with several different molecules. We really have an unrivaled publication record, and you'll hear about some of those as case studies. The ProPresent assay is essentially a two-step process. The first is the generation of cells that have come in contact or been exposed to an antigen in some form or the other, whereas the second step is the recovery of the peptides and sequencing of those by mass spectrometry. Now, we can generate the cells in-house like a dendritic cell and load exogenous material, and again, that's what Jeremy will be talking about, or we can work with whatever type of cell that you provide to us. And I think what speaks to the versatility of this assay is the cell source and the antigen sources that are listed here. These can include tumor cell lines, cells from healthy individuals who have received some type of vaccination, or cells from patients who've received some type of treatment, maybe an RNA or DNA-based vaccine. Once we receive those cells, we'll then do an immunoprecipitation step, pulling down the MHC peptide complex. Those peptides are then recovered using mass spectrometry, and we'll create a database that contains the human proteome and that sequence from the protein of interest. We'll then report all of the peptides that are presented from that peptide. We can also look for mutated sequences, so if you have tumor tissue and you've done the exome sequencing, we can search for those neoepitopes, as well as other peptide modifications like phosphorylation. Okay, so the first case study comes from the Children's Hospital of Philadelphia, and this is the use of the ProPresent assay to identify peptides from their adeno-associated virus vector in their gene therapy program. Now, AAV is a very commonly used vector. However, there is a very large portion of adults that have neutralizing antibodies against serotypes 1 and 2. So it's very important to monitor and understand what the immune responses are against that virus. So the purpose of this study was to identify AAV peptides using ProPresent so that that could be implemented into their immune monitoring strategy. So they provided us with HHL5B7 hepatocytes that had been transduced with an AAV2 vector encoding the capsid alone or human factor IX. And then we performed the ProPresent assay, and that data set is shown here. Here are their 2 sample types, followed by the sequence that was presented from that sample, the protein of origin, the amino acid start and end position, and the expect value, which is similar to that of a p-value. You can see there's one sequence, this SLD, that's highlighted from both sample sources between amino acids 429 and 437 that also had low expect values. So this was indicative of an epitope, and they went on to show that there were functional CD8 T cells that recognized it using an interferon gamma ELISpot. The second case study comes from the German Cancer Center and Oryx, an oncolytic virus company. And this is the use of the ProPresent assay to identify peptides from their parvoviral therapy. Now, oncolytic viruses are very promising cancer immunotherapy in that these virus will selectively replicate in the cancer cells to induce an anti-tumor immunity. And you can enhance that immunity by providing it with stimulatory genes or in combination with something like a checkpoint inhibitor. There are several different oncolytic viruses that are being explored, and in this particular study, they were looking at rodent protoparvovirus, which is non-pathogenic for humans, very small, highly oncoselective, and it causes direct oncolysis. They were looking at their PARV-ORX-01 treatment in a phase 1/2a trial for glioblastoma multiforme, which is the most common primary brain tumor. It makes up 2 to 3% of all human malignancies and also has a very poor prognosis with 50% of patients who do not survive past 16 months. So very clear need for a very focused treatment. The objective of the study was to identify relevant parvoviral and glioma epitopes that could be used for their immune monitoring portion. Now, they had very few cells to work with, and those were reserved for immune monitoring. So for the epitope discovery portion, they got very creative with their solution and provided us with glioblastoma cell lines that had been matched to the patient's HLA type and then infected with parvovirus. So they provided us with those cells. We did the ProPresent assay and identified 5 unique peptides from 2 different parvoviral proteins and 20 peptides from 18 different glioma proteins. We then went on to do the immune monitoring portion of the study for them. We used a T-cell ELISpot assay. So before I get back to the case study, I do want to introduce this assay briefly. It's essentially the assay standard for looking at T-cell immune responses and confirming T-cell epitopes. It's a very robust and highly reproducible functional T-cell assay. It measures cytokine production at the single cell level following stimulation with an antigen. It's quite sensitive, and depending on the ELISpot handler, you can detect responses as low as 1 cell in every 100,000 with a low background. We typically assay for interferon gamma, but you can see here in this list there's several other cytokines that can be looked at. The antigens can include a whole protein as well as individual peptides or peptide pools, as we heard earlier. And those peptide pools can contain a handful of maybe immunodominant HLA-restricted peptides or an overlapping peptide library. We use whole PBMCs, which encompasses both the CD8 and CD4 T cell responses. Of course, you can further purify the cells and use the appropriate length peptide, should you want to look at one or the other. And then probably one of the most important aspects of an ELISpot is the cell quality. It's very important to process the cells in a very timely manner following blood draw, and that a robust cryopreservation technique is used. So I'm going to be providing some examples of different optimization and qualification studies that we can provide to achieve these criteria prior to ever working with your clinical samples. So we do perform an overnight ELISpot using cryopreserved whole PBMCs. We plate 125,000 cells or 250,000 cells per well in triplicate, and following the overnight incubation, we'll then count the spots using an automated plate reader. So this can be a cell-intensive assay based on the number of antigens that you would like to use. We will always include, in the form of 3 antigens, our controls, which include PHA, CEF, and media-only negative control. Okay, so back to that case study. In the parvo-ORX-01 treatment, there were 4 antigen pools that were used, 2 from glioma and 2 from parvovirus. 3 pools contained novel epitopes identified by ProPresent, and 1 pool contained peptides taken from the literature. Okay, so here are a couple of the patient data. What we're looking at here are the number of spot-forming cells per million, PBMCs, and on the x-axis are the time points post-treatment. At day 61, you can see that there's significant responses to both positive controls, which we like to see, but there are also significant responses to all 4 of the conditions. And again, 3 of these 4 peptide pools contain novel sequences identified by ProPresent. With this other patient, there's significant responses at day 18, 57, and then also sustained significant responses for the parvovirus pools at day 120. So ELISpots have been incorporated into clinical trials more and more, and as you can see, we have a lot of experience in this area. We've worked with the Oncolytic virus groups. We also have experience working with gene therapy groups who are tracking responses to both the viral vector and the transgene. We have completed a trial working with a group and also are currently working with one on their clinical program right now. We've also worked with a clinical program for Immune-oncology, looking at an ADC candidate, and are currently working with Celgene on their CAR-T program. We've done epitope mapping on several different types of viruses and even done some work on immunotherapies for cat allergy. So we have an abundance of experience in this area, and what we'll do is provide insight on how to optimize the study for your particular project. And we'll do that by beginning to help you understand what the limitations of your own study are. So the number of antigens that you can include in your assay will be directly dependent on the number of cells that you have to work with, which will be dependent on how much blood is being drawn from your patients. So if you're working with pediatric samples, for example, and you're limited to just 4 mLs of blood, you'll have very few cells to work with. So once that's been determined, we can help design the peptide antigens. Again, we have experience in making a lot of different types of peptide pools, including AAV, the transgene, the CAR-T constructs. We'll also do the pooling and aliquoting for you. Once the peptides are ready, we would recommend beginning with identification of a positive control donor when possible. And to do that, we've used the ProImmune PBMC Biobank, and these are PBMCs that have been collected from healthy individuals. They've been fully typed for class 2, and then we'll select a panel to screen an ELISpot assay against your desired antigen peptide pool. So we have done that for example, one of our gene therapy clients, and this is the data for that positive control donor over 10 different ELISpot runs. So in green, which actually doesn't show up on the scale at all, are the media-only responses, and they're consistently very low, so very nice low backgrounds. We see nice responses to PHA and CEF, and the nice consistent responses to the antigen peptide pool as well. And with this particular donor, this played a very important role because they were having issues with the functionality and viability of their clinical samples. So by seeing how this donor responded in the assay, we were able to help tease out some of the issues that they were having with their clinical samples. Which brings me to the next qualification step, which is that of any third-party cryopreservation protocol. So the blood is being collected all over the world for these clinical trials, and that isn't necessarily where your cells are being processed. So we'll work with those multiple sites to establish a cohesive protocol for both processing and cryopreservation, because the tiniest of detail can make a major difference in your cell's viability, whether that's the tube type that's being used or how quickly the cells are spun down following processing. And one thing we would recommend is having those sites process cells from a healthy individual, sending those to us. They make the trip over the Atlantic, and then we can test those in an ELISpot assay with the peptides that will be used in the actual trial. We'll also help to establish a prioritization schedule. So when we go to thaw the cells, if there are not enough available to run the scope of the study, we'll have a predetermined plan that we can follow, whether that's to reduce the number of replicates or to eliminate one of the controls. We can also reduce the number of cells that are plated per well. So with some of the groups, we've actually done cell limiting studies to reduce the number of cells from 250,000 per well down to as low as we could go to still see a response against their antigen. And then that gets incorporated into that prioritization schedule. And then the last type of qualification study that we would suggest is an inter and intra-assay variability study. And we first performed this when we launched the ELISpot assay. And this is the results of 2 different ELISpot handlers over 3 days for a total of 6 ELISpots against test peptide A and B, as well as a negative and positive control. So we did see consistently low levels of background responses as expected, less than 20 spot-forming cells per million, and then consistent results against each of the test antigens over those 6 ELISpots. So this report is available, and again, we would suggest doing this with your own peptides. So once those qualification studies are complete, you can have confidence that we will be receiving cells that are of the highest quality and functionality, and that we have optimized the assay to fit both your antigen and your population cohort. The cells typically come to us as batches, so we'll run each batch as they're ready, and then we'll provide the data report in about 1 to 2 weeks following receipt of the cells. So if you'd like to hear more about how these types of optimization studies and ELISpots themselves have been incorporated into clinical programs, Dr. Priya Sriraman was here at this conference last year and provided an excellent presentation on ELISpots and their CAR-T program. So I encourage you to have a look at our website. You'll find her talk, as well as many others, and then the talks from this conference as well. Okay, the other type of epitope discovery tool that I wanted to describe was our MHC-peptide binding assay. Now, this is a high-throughput physical peptide binding assay, so it does help to eliminate some of the inaccuracies that can be associated with in silico approaches. It is high throughput, and it rapidly identifies the precise HLA restriction of T cells, the T cell epitopes. Also, one of the important features is that it doesn't require any cellular material, so it can really get incorporated at any point in your research. It's a modular platform, and for Class 1 we have 4 modules. The first being the synthesis of peptides, the second being the actual binding assay itself. So we'll combine your peptide of interest with an MHC molecule and the beta-2M in the presence of a conformational antibody. And that antibody will only bind to the MHC peptide complex given the appropriate configuration. And when that occurs, a fluorescent signal is generated, and we'll use that to create a binding score relative to a known T cell epitope from the same HLA allele that we run in parallel. Anything that's considered a strong binder, we can then synthesize proved pentamers, which are flow cytometry reagents to detect antigen-specific CD8 T cells, and I'll come back to those in just a moment. And then the fourth module is a more detailed characterization of the stability of that peptide, measuring the on and off rate, or the kinetics. So the next case study comes from the Imperial College of London, and this is the use of the REVEAL binding assay to identify novel T cell epitopes for HIV. And with HIV, it's well known that the HLA type of the individual plays a major role in the progression of the disease, and that B35 expressing individuals are associated with a rapid disease progression. And much like other viral diseases, it's known that the CD8 T cells play a very important role in controlling the infection and subsequently clearing it, and that one of the major targets is HIV-GAG, and the higher the response against HIV-GAG, the lower the viral burden. So the purpose of this study was to use the binding assay to determine B35 restricted HIV-GAG epitopes. So they did an initial screen using the Immune-epitope database, screening the GAG sequence, and they narrowed down to 44 peptides that they then had ProImmune run in our REVEAL binding assay. And the results of that are shown here. They ran this against a pass-fail control, and everything that was of 80% or higher compared to that pass-fail control was considered a hit. From there, they then went on to do the quick check analysis. So we heated up the MHC peptide complexes to 37 degrees and then measured the off rate at 0, 2, and 24 hours. We then used those half-lives to determine a quick check score, which is what you see here on the y-axis. And you can see there are two peptides, number 1 and number 8, that had very strong quick check scores, indicating they're quite stable. So from there, we synthesized 2 pentamers for them. Again, these are our multimers, and it's a flow cytometry tool. And that pentameric structure is shown here. We have 5 MHC peptide complexes configured through a coiled domain with 5 fluorescent tags. And those fluorescent tags can be APC or PE, or unique to the pentamers. You can get these with a biotin label. So this obviously gives you the flexibility to incorporate it into any type of staining panel, as you can use streptavid in whatever color you choose. Also unique to the pentamer is you can get these without the fluorescent label. And these are really nice because they have an extended shelf life of a full year. So you can imagine if you're working with clinical samples over a very long period of time, this will allow you to order a large amount of pentamers from the same lot, limiting your lot-to-lot variation. You receive those, you do all your initial QCs, you aliquot them down and freeze them, and then use them as needed. So you go through much less of your cellular material that way. We have over 450 catalog items available, from which more than 200 are cancer-specific, for example. And if we don't have your specificity available, we can also do custom synthesis, which takes about 4 to 6 weeks. And then what speaks to the consistency of these reagents are the number of scientific publications. We have more than 1,600, and those relative to what I've been talking about today include 14 that are pertaining to adeno-associated virus, 15 with vaccinia virus, and 232 that are cancer-specific. So again, we made pentamers for those 2 epitopes, number 1 and number 8. This is the data for number 8, this HPVH sequence. Shown in the upper right quadrant are the HPVH specific CD8 T cells, which is 0.39%, so a nice clear population. Okay, the last case study comes from the University of Pittsburgh, and this is the use of the REVEAL binding assay to identify HHV8 epitopes. So as Jeremy mentioned, this is the project that I was involved in. Human herpes virus is the causative agent of capsucsarcoma, which is the most common AIDS-associated cancer. And of course, one of the hallmarks of a herpes virus are the latent and lytic life cycles. And with this particular herpes virus, the lytic cycle proteins are known to contribute to the angiogenesis of the tumor, or of the endothelial cells where the tumor is derived. And that CD8 T cells play a very important role in controlling the life cycle of that virus, especially the viral proteins. So the purpose of this study was to identify the latent and lytic cycle peptides that could serve as biomarkers for the individuals who had the virus that did or did not go on to develop the cancer. So I mentioned that ELISpots can be used as an epitope discovery tool earlier on, but that can take a while. So both myself and the primary author for this project spent many months doing ELISpots on several different HHV8 negative and positive donors over several different viral proteins. So this is a great example of how the REVEAL binding assay could have been incorporated earlier to reduce some of that time. But we were cheap labor, so we just continued to do those ELISpots. And then we came to ProImmune with 10 novel epitopes and 4 that had been previously published. And then they ran those in an A2 binding assay, and that data is shown here. Everything denoted with a star was considered a strong binder. And from that, they synthesized pentamers for us, and we did detect significant percentages of LANA-specific, which is a latent protein, and K8.1 specific CD8 T cells in the HHV8 seropositive individuals. Okay, oops, I do have one more. So this is the last case study coming from Oxford Biomedica, and this kind of encompasses all of the different types of assays I've mentioned. This is the use of the MHC-Pentamers to validate novel CD8 T-cell epitopes in colorectal cancer patients vaccinated with Trovax. And Trovax consists of a recombinant vaccinia virus encoding the tumor-associated antigen 5T4, which is expressed on high levels of solid tumors. So we first synthesized a peptide library for them, which was used in an interferon-γ ELISpot to validate epitopes that had been identified in a binding assay. And that data set is shown here, both pre and post vaccination. We also synthesized pentamers for them, and that data set is shown here. Here is pre-vaccination, and here is post-vaccination. So a nice percentage of 0.22%. Okay, so all of the assays and reagents I've shared thus far have been geared towards class 1. They are also available for class 2. And we have class 2 tetramers to determine and quantify antigen-specific CD4 T cells. And for that, we have over 132 catalog specificities, several antigens that are listed here. If your specificity is not available, we can also do custom, just like with the pentamers. These come with either an APC or PE label, and those custom tetramers are delivered in about 4 to 6 weeks. Okay, so lastly, I just want to introduce our newest reagent. This is something we just launched last week. Yeah, these are MR1 tetramers, and these are used to detect mucosal-associated invariant T cells, or MATE cells. And MATE cells are an unconventional T cell that resides primarily in mucosal linings, like the gut, but also make up about 10% of your blood T cells and about 45% of your liver T cells. And they have conserved TCRs that recognize the intermediates from microbial synthesis of vitamins. So these intermediates are not your standard antigenic peptide, and they're not presented from classical MHC molecules, but rather by the MHC class 1-like protein MR1. So we've recently launched these, and you can get them empty, ready for loading, preloaded with that intermediate or with a negative control. These are also PE and APC labeled in various test sizes, and much like our other products, they're guaranteed for 6 months. Okay. So in summary, to have successful epitope discovery and immune monitoring programs, it's important to have a very detailed and efficient characterization of the antigen-specific and anti-tumor epitopes. And it's important to have access to a wide range of these specialist assays and tools to enable the appropriate decisions are made in a very timely manner. And the type of data that I showed you today and the generation of that data will help to differentiate your vaccine or your immunotherapy from your competitors. So we have a wide range of experience in these areas, and you got to hear a little bit about those research areas today, and then you'll hear about many more of these types of molecules tomorrow from Jeremy Stock. Thank you very much.
Any questions?
Sorry.
Thank you very much. I have a question regarding your qualification studies. Do you have any additional, did you do any qualification studies inter-assay, intra-assay for other assays that you have? You mentioned the one for the
ELISpot assay.
ELISpot.
Yeah, that might be something that, yeah.
Thanks, Sophia. So it depends a little bit on what the assays are. And obviously one of the challenges with some of the assays that you're probably thinking about are the fact they're pretty cell intensive. So where you have a limited numbers of cells available, to kind of reproduce those with ends of 10, 12 is essentially impossible with the numbers of cells that are available from individual donors. But we do have obviously evidence for a lot of the different studies where we have done a certain amount of intra-assay variability, the confidence of the data that we're generating. So for one example, in the antigen presentation assays, we know where we have had some situations where we've actually been able to source blood cells from the same donor at different time points. That was only in one case. So the data set is limited, but where that was the case, we identified the same peptides. Well, I say exactly the same. I mean, it sort of overlap about 70% of exactly the same peptides being identified, but that was several months later. So obviously the blood sample was taken after maybe their exposure to antigens was also different. So I think given that's the case, then it's pretty good reproducibility. But it does depend, of course, on the different assays. And Nick's got a comment as well.
So I think as you heard from Emilee, we do these sort of qualifications, let's say, for the ELISpots, usually in the context of the customer's objective, because without that context, you can't really qualify anything. So here for the ELISpots I've worked, an objective is often to qualify whether the cells that we're getting from the actual clinical sites perform in an ELISpot assay against the control responses. So all the other types of assays like T-cell proliferation assays and so on, then there's a question of what is the objective exactly of what to measure. I mean, you've got basic control responses, epidemiology and ALHs as sort of absolute positive and TB as well. So we have more sort of longitudinal data to show that we get stable responses on those. If you have a specific drug in mind and a specific objective for you want to measure out that assay in the context of that drug, then the qualification study will essentially have to be designed specific for that objective and not to prevent.
I just have more of a high-level question on, we know that the guidance on the gene therapy and the cell-based and nucleic acid therapies are now being formulated by the agencies. And so the immediate need is for development of humoral assays, mainly the antiviral immune responses or even the transgene-specific antibody assays. So what is the, have you seen a lot of, working with your groups, a lot of requests which have triggered the need for these cell-based assays? Because there is a limitation to how much amount of blood you can get from the patients. And then, of course, there is, giving an example of oncolytic viruses, yes, you can look for the viral capsid proteins or even the transgene, but sometimes the transgenes are intracellular. And so what is the, why would you develop a transgene-specific cell-based assay if you're never going to expose it to the periphery? So are those things thought about when these kind of assays are developed and considering the amount of sample resource it would take?
Yeah, no, that's an excellent point. As far as the humoral aspect, yeah, we haven't, I guess, done much with groups on that basis. But for the cellular aspect, yeah, that's something we take into consideration because it kind of gears what your antigen pool is going to consist of. Because if you only have enough cells to look at a couple antigens, then we can put your viral vector in one, we can put your transgene in the other. But if it comes down to it, I suppose more people are going to follow the responses to the vector than they are the transgene, independent of where it's expressed. That's it. Okay, thank you.